That makes sense. That's what journals.
And we had a blood test and we had
No, I understand that most of us you're a psyche up for when you have these
I
figured I could definitely make a fellow, even the fellows said no.
So again, we're out in the January journals. I'm out.
You're out of that and do it this, what was it, 17?
Yeah.
Because I didn't mention that.
We have
Well, they have actually
last week.
Going later in the year.
Projected correctly. When they were video, when people outside were watching, they were watching a video on the speaker screen, so they couldn't see any of the slides. It's because the resolution changed just a slight bit. It's just crazy. I would never have thought that.
And there's more of one type in Japan and how you are in other.
Oh, it is.
It's really complicated. You have to do study.
So sometimes we're giving you a shirt.
it's not journal club, it's journal.
Oh,
Overall, compare
For some reason, it's coming up.
I know it is tough, especially in science. It's so complicated. No questions. Very good. Review journals. That's your quote stuff. That's right.
So this will be.
you know, the first of its own.
Yeah, that's right. On science, I don't read that.
I mean, I think it's going to be.
So Paul has two. This is Kevin's. Here's Kevin's. And Matt can help with this too. I'll bring up his talks and if Amy can't get
So,
him. Yeah, that's up to him. He can come up.
he'll just have to tell me which one it is.
Yeah.
And
Matt.
so he'll do that.
Well, I signed up for a trip, but it's over a year away. You know, you gotta sign up for it.
Maybe you might, yeah, I was going to say, you might want to.
Try to get home for next week. No, I'll be back.
No, I'm still working. I'm supposed to be away.
I was up.
Doctor, I have inflammation.
I'm busy.
Can you hear me?
I had that.
I have inflammation.
I have inflammation all over.
It's all over. I mean, it's like fixing my tissue injury. It's you gotta stop this stuff.
No, I want to go to Angkor Wat. There was another one that went in Cambodia. It was on the coastline. And I didn't realize Angkor Wat's way out.
Oh, it's in inland, yeah.
And I did this like, you know, just drive
It's all marshes.
That's why I decided to trip to Cambodia, Vietnam, Thailand. I'm going to do the land trip on that one.
I sent
Fruit.
I sent hey Drew. I sent you an email about 15 minutes ago. Did you get that?
I haven't checked it.
You've been in Australia, New Zealand, right?
Just barely. I'll tell you what, it's a bit. I've been in the North Island. I did that fairly well. And dropped it off and rented a car, drove around the North Island. So I had to the Southern Island where all the and the other place I just visited, the cruise. And so we had a day in Sydney, and I saw a lot of cities from that place, but I haven't been up to the great. I want to go to the car.
Yeah. That's one of our good trips.
I'm trying to do one every year. Actually, I'm signed up for 2 next year. They have a nice cruise, and it's areas that we have done. It goes to the Norwegian fjords, it goes to Stockholm, it goes to
I was so thankful to get out of like this. I'm like, oh, XP is so nice.
Berlin, Berlin, and Berlin, yeah, you know, to date, and so, and I haven't done, I've been to Norway, I've been to Sweden, I've been to Stockholm, I haven't been to Estonia, and I haven't been to Berlin, and I have never been to
They created around elevation, leaky gut, anti-inflammatory diet. All of this is all part of like this tightly wound package.
You know, they charge you so much more for this higher.
And all the like the tests, like the HLA testing.
That was even
What line?
that was Viking. Starting since
We have celiac disease. So we have non-celiac HLA positive gluten sensitivity.
Now we're in phase 1 in Daniel, but test for line C. And then we did China.
Yeah, I got the very end of that story. No, so what's the
rationale is
That'll do it.
But as soon as I got ready to sign up, well, the cheapest one.
Don't you want the best for your baby? We're going to use sciencey tools to. I try to remind myself that being gluten sensitive is its own punishment. And we don't want that.
Yeah, really.
So maybe they're doing us a service. I was going to an ancient white person. What's that?
Yeah.
Product great. AWS.
One of our speakers isn't here, but
Who's missing?
Paul McBride.
Oh, Paul, he's
But he's got
holy smoke. He's all reliable at traffic.
Yeah.
I think I saw that. It was a white station wagon.
no, that's
Oh, it's like a Jerry.
I wanted to
it's got BMW. I think. You drive my chair.
Yeah, but mine's so.
Good morning, Dr. Guilan. Pleasure to see you.
One of our speakers isn't here, but let's go ahead and get started. Today's journal session. One announcement, well, the usual announcement to sign in for CME credit, the word of the day is inflammation. The last session of the year is December 17th, which is scheduled to be Paul Williams on, I don't know what his topic is, I forgot. But anyway, yesterday he asked me, he had something that was interfering with that, so he asked me to change, so we're going to change to the journal session in January. So we'll send out a revision to the schedule. You might just want to make note of that. The last session stays, the date stays the same, just what we do that day changes to a journal review. And Paul will do his subject, whatever date it was in January. You remember, Gary, you're the one who's making one of the
You
swaps.
sent me the email, but I didn't pay any
All right,
attention.
anyway, you'll be aware of that. So today we have three speakers, myself, Kevin, and Paul McBride, if he gets here. I just have to assume the fog is holding him up. So I've got three papers that I'll try and go through pretty quickly. I'm glad that Matt is sitting here because I'll probably need his help in getting through the slides. Good. The first one I have is about a device that I learned about that I've never heard any of us talk about in this room, so I figured maybe no one else was aware of it either. It's a device that the ENT guys have. It's a resorbable steroid eluting stent that they put in at the time that they do functional endoscopic surgery. And it's already FDA approved to use at the time that they operate. They're doing a subsequent study now to try and get permission from the FDA to use the exact same product in the office. I should back up and say we've had so much basic science recently. I Just going to do some very simple-minded clinical medicine, which once in a while it's good to do. So this is one of about 4 or 5 papers in this field just showing the data that got this product approved. I don't have any disclosure. So this was a prospective randomized double-blind study in 43 patients from, I think, 3 different ENT groups in the U.S., where they placed the active stent in one location and on the opposite side, stent without any steroid. It was double-blinded, so all the observations were made by ENT surgeons who didn't know which side the stent, the active stent was, and then they followed these people endoscopically for 60 days, looking for inflammation, which was just clinical impression, although blinded on a visual analog scale of 0 to 100, then polyp tissue reformation, adhesions, and turbinate position. And basically, the results were quite simplistic here. The active side reduced inflammation between the window of time, day 21 to 45, with statistical significance, reduced the redevelopment of polyps and the frequency of adhesions. And here's just a little bit of the data out of it. This is the percent of the stent. It's a bioabsorbable material. It's roughly the same material you use in sutures. And it disappears almost completely by day 30 after it's put in, and it's totally gone by day 60. And this is just some of the inflammation data. Again, no hard objective data like biopsy or anything of that nature, but just visual impressions by the ENT people. But again, at all the points where the stars are, there's statistical significance that there was reduced inflammation. This was supposed to be a video that I took off the internet, which I think is lay advertising for the lay public. Advertising is kind of a slick video, but we can't get it to play. But that's a magnified picture of what this thing looked like. I've held one in my hands. In reality, it's about the size of the tip of your pinky. And they can physically put it into the surgical wound, and then it expands to fill the space, and then it elutes steroid. Most of it's eluted out. It's mometasone, 370 micrograms of mometasone that's embedded in this, and it just elutes by diffusion over about the first 30 days that it's in place.
What's the benefit of this over just spraying stuff in the nose?
Well, they make the point that, you know, most of the time the stuff that you spray doesn't get where you want it to go.
Yeah, but if they did those same scores, they'd have the same results.
Well, if you're criticizing the quality of the science here,
Or the practicality.
Yeah,
The practicality
I mean-
more than the science.
I mean, all I can say is these guys are using it. There are surgeons locally who are using it. They have about 4 papers of about equal quality in the literature, all in the ENT literature. And I just thought I'd make people aware of it. I know there's at least one surgeon locally who's doing it, because I sent him a woman who's got just terrible sinusitis, and I was trying to manage it medically without success. And he's going to place it. And there is, as I said, a study. So they have FDA approval now to place this at the time of surgery. Technically, it's very simple. I've talked to a couple of guys. They want to get approval to just do it in the office. The closest site where they're doing a study to get the permission for local in-office is down in Oregon Health Sciences. Nobody here in Seattle is studying it. So basically just an FYI that this device exists, and we may see our colleagues in ENT using it.
Greg Davis, I know.
Is doing
Because
it.
I've had 3 patients that desensitized, to aspirin, and they've all had this in.
And do you have any impressions of local use of it?
Not really. I mean, it's hard to.
So you have to keep putting it in every 6 weeks or something?
No, I think it's a one-time procedure.
I mean,
The concept is if you get the steroid locally to the diseased tissue right after the surgery, and you reduce adhesions and polyp formation early in the wound healing process, your total outcome will be better. That kind of data they don't have. It's really what the long-term regrowth of polyps is. Anyway, just a heads up. And then I've got two papers. After having Dave Golden here, I thought we'd just talk a little bit more about venom immunotherapy. So in our new journal, the In Practice Journal, which I've actually been impressed is a remarkably good journal for something that launched so quickly. The most recent issue just came yesterday, and it's really, it's more clinical, but as far as the quality of the stuff in there, it's really very good. So this is a paper on venom immunotherapy in patients with clonal mast cell disorders, as you can read, efficacy, safety, and practical considerations. And the reason I selected this was I was really impressed that any group of people, it's actually a large group from both Spain and Italy, had so many patients with these two conditions together. I mean, I've seen it like once, where I've had to deal with a mastocytosis in a guy with venom anaphylaxis. So they have 84 patients with this condition. So they had, clearly the largest group I would think in the world. Prospective study in a group of people in Italy and Spain over 13 years. You can see the age of the patient. One thing that they don't mention in great detail, but a lot of their people are beekeepers, so that's how they get such a large group of people. But they have a sizable number of people who also are not beekeepers and are sensitized to other Hymenoptera. So they all had a systemic reaction after a sting, an elevated tryptase or urticaria pigmentosa. They all had bone marrow biopsies to look for mast cell atypia, CD markers, and the c-KIT mutation. And they worked up them by traditional methods that we would all use, skin testing and measuring venom-specific IgE. Different than what we would do here, I think, for the most part, is their treatment schedule. They broke into three different ways of increasing, or the induction phase of immunotherapy. Conventional 15 weeks, one dose per week, the way it's pretty much done in the U.S. by most of us. A modified brush where they cut it in half and use a 7-week schedule, and then weekly they would just increase, skipping the first 7 doses, and then a true rush schedule where they got people through the entire advancement in 4 days and just clustering doses on each of those 4 days. And then when they got to the 3rd year of maintenance, they moved people out to every 3-month maintenance, which is actually the subject of the next paper that I've got. So this is just the demographics of the people in their group. They had indolent systemic mastocytosis with skin lesions, without, monoclonal mast cell disorder. So the N and the percentage in each of those categories, the mean tryptase of the group of individuals, and then the CD25, CD2, and the c-KIT abnormality, just outlining the data in these people. The severity of the reactions before they put them on venom immunotherapy, really quite severe, most of them with a grade 4 reaction, and a very high percentage with loss of consciousness. And as we know, actually, this group of people who have mast cell disorders and venom reactions can have a high risk of loss of consciousness and a much heightened rate of severity of reactions and increased concern about fatality. The breakdown of the people as to what they immunized them with, not just B, but Vespids, wasp, or a combination of Vespid and wasp. So the point of this whole thing really is it safe to put people with mast cell disorders on venom immunotherapy, and is it effective? And the outcome is yes to both of those. They had 10 adverse reactions during the induction phase in 10 patients, but 6 were large local reactions. So actually only 4 of those reactions were truly systemic. They all occurred within 30 minutes of giving the immunotherapy injection. So thus, as I mentioned, only true systemic reactions in 4 people, and only 1 of them needed epinephrine for treatment. Interestingly, 3 of those reactors were in the conventional, slower advancement. 7 were modified, and in the rush So even though they're rushing these people, admittedly it was a small N, they had no adverse reactions. They had no adverse reactions at all during the maintenance. 60 of the 84 people, they kept on venom immunotherapy for at least 5 years. So adherence to injections was 88.4%. That was their other point here, that they feel that, as they talk about later, stretching out maintenance to a longer duration keeps adherence to a higher level, hence saves money for society.
Yeah, but they do versus our sort of typical patients. Anything atypical in terms of mediator blockade, like really high doses of H1 blockers.
For pre-treatment?
Yeah.
Yeah, no, they didn't mention
They didn't mention it.
and there was no difference in mean tryptase levels in those who had adverse reactions versus those who did not. Then they had a certain subgroup of those people had restings. So 50 of them, half of them had a field resting during the maintenance phase, a total of 95 stings in that group of 50 while they were on maintenance between 2 months and 7 years. 7 of the 50 had reactions to those restings, and their response to that was to increase the maintenance dose to 200 milligrams, and only 2 of them have then a second subsequent resting with no reaction. So their overall conclusion is that venom immunotherapy is well tolerated, safe, and effective in patients with clonal mast cell disorders. I'm just curious if anybody here, I have had one patient with this combination of 2 diseases. Do we have any other experience in the room? No.
Not that I know of.
All right.
Probably do.
We probably do and miss it, is the thought. The last one is something we just quickly reviewed in our practice here, because this has been a controversial point. Can you extend the maintenance of venom immunotherapy out to a longer window of time? And the bottom line, basically based on this paper, is yes, you can. And in our practice, we just recently agreed to do that, to be able to move people's maintenance phase out to every 3 months, although we had disagreed about that in the past. So it's more or less actually partially the same group of people in Italy. This was a prospective observational study of single venom immunotherapy, comparison of conventional versus extended maintenance, extended being at one point out to 3 months, later out to 4 months. Again, different than the way we would usually use venom immunotherapy. They did a rapid buildup, they did a rush phase over 3 days, a week apart, so they got them up to 100 micrograms of honeybee or yellow jacket in a very quick period of time. And the extended group, then they pretty quickly extended maintenance out to 6 weeks, then 2 months, then to 3 months. They actually had them out to 3-month maintenance by 4.5 months into the entire treatment phase. And then at the 5th year, they decided to extend it out to every 4 months. Now, this is just the data. So EMD means extended maintenance dose. CMD means conventional maintenance dose. The only thing I want to highlight is just these were not matched groups really very well, because they started the conventional dose at a different point in time. So if you look at the actual total duration of venom immunotherapy, it's much longer for the extended group. And I'll come back and just make a comment about that at the end. So again, quickly here, it's a busy table. If you just look at reactions before immunotherapy and then during immunotherapy, essentially what you see here is that people who had high reaction rates before had virtually no reaction while they were on immunotherapy in both groups, whether they had conventional or extended, true of honeybee and yellow jacket. So their actual summary of the data is that in restings, so basically the point was here, they put people on extended dose, then followed what happened when they had restings. Restings without reaction were 93.5% in the extended group. That should be 81.5% in the conventional group. So actually fewer adverse reactions to the restings in people who are on extended maintenance dose. No difference in reactions when they extended it to 4 months. And then because of that mismatch with the total duration that people were on venom immunotherapy, they reanalyzed their data a subgroup of people who were just on the extended and the maintenance dose for an equal period of time. And their re-reaction rate is, as you can see, was low in both and not difference between the two. So that's all of that presentation. So basically, I think, again, one of the better studies, larger groups, showing that we can extend venom immunotherapy maintenance out to 3 and ultimately out to 4 months. So unless any questions, Kevin's got some papers and Paul has got some as well.
I tried to go for a theme, and the best I could find was egg allergy. So the first paper is early regular egg exposure in infants with eczema, an RCT. This is building off the most recent feeding guidelines, which say don't restrict any particular foods for babies at 4 to 6 months when they start solids. So the question this group wanted to ask is, going the other direction, what if we deliberately gave high allergy food to young babies? Could we maybe reduce the risk of allergy? So this is a prospective trial in Australia, and looking at this intervention time between 4 and 8 months of life, they gave kids either an egg powder or rice powder, and this is a pasteurized egg, so it was safe in that sense. It's about a gram or a sixth of a total egg, and it was mixed into like a rice cereal. They gave it every single day, and for both groups there was no other egg exposure. They didn't talk about mom's diet, whether the babies were breastfed or not. I would assume that was probably another source of egg exposure. At the end of this intervention, at 8 months of age, all the babies were exposed to cooked egg. So they had around 2 teaspoons of mashed up boiled egg. And then 4 months later, at a year of age, they were all skin tested to egg, and then there was an oral food challenge. There's a couple of babies who had some anaphylactic events. We'll talk about that. They were not included in the challenge at a year. So primarily the first outcome was to see what was the rate of IgE-mediated food allergy to egg at 1 year of age. That was defined by both positive challenge and skin test positivity. There's a number of secondary outcomes, including IgE and IgG.
These were high-risk kids who already had eczema?
Very high risk. I forgot to mention, so these were babies who had moderate to severe eczema. So they really stirred the pot here. We'll see that it was maybe not the wisest choice. So there were no differences in the baseline characteristics, age, gender, and so forth. Among the babies who were randomized to the egg powder, so the kids who got a teaspoon of egg powder every day from age 4 to 8 months, 15 had allergic reaction at some point during the process. 10 of those 15 reacted at the very first time they were given egg, that 4-month mark. So they actually had become sort of been pre-sensitized, if you want to say that. There's a case of anaphylaxis in there. It's a very, very risky group. Even in the rice powder group, the control group, there was still some reactions, some hives, and maybe some vomiting. I can help but wonder if there's maybe some FPIs going on in that group. And they actually left the study, it was so bad. So, not great compliance in terms of egg avoidance for the rest of these babies. A lot of them had egg exposures accidentally and sometimes repeated egg exposures. There were 4 serious adverse events, including FPIs in one baby to the egg. This is a baby who had probably some vomiting symptoms, and they went and re-challenged and then drove to the emergency room for FPIs, and then 3 anaphylactic events to egg. So almost a very explosive group of kids here. As a result, though, they had to go back to the study board, and there was a delay. Their funds sort of got weak, and then they had poor enrollment. And so they had to use an intention to treat analysis, which basically says we're going to use the data we had and compare it against our goal, which really weakens the strength of the study, but that's all they could really do. So looking at the primary outcome of the rate of IgE-mediated egg allergy at 12 months of age, 33% of infants in the egg powder group, so ones who got egg powder every day from age 4 to 8 months, had, they called it a trend. Couldn't really prove it statistically, but there was a little bit less allergy in that group compared to the control group. Secondary outcomes looking at just positive skin test to egg at 12 months, also no different between the two groups, although I want to talk about the trend again. In terms of eczema natural history, these kids had no differences in their eczema control, amount of steroid use, and so forth. Looking at egg-specific IgE, at 4 months of age, when they all enrolled in the study at the very, very beginning, there was a higher rate of already being sensitized to egg at that point, so 36% already had measurable IgE. Not surprisingly, if you took the IgE levels, they correlated with the rate of egg reactions later in the study. And then specific IgG4 to egg was elevated in the patients who had persistent egg exposure. The kids who had more egg basically had higher IgG4 levels. I'm going to skip graphs for time here. So the take home here is this is a very risky trial in retrospect. You're taking very, very high risk kids, so moderate severe eczema, and exposing them to egg. They found a lot of sensitization early in the study where kids hadn't even been exposed to egg theoretically, maybe through breast milk or their skin or some other means. They had a lot of adverse events which caused huge disruptions in the study and probably was a major deterrent to their ability to interpret the data. In their words, though, their conclusion was that early frequent egg exposure can induce immune tolerance pathways and reduce the incidence of egg allergy, which really sounds more like a hypothesis than a conclusion from the study. Because they really couldn't draw any conclusions, in my opinion. Although they said basically we didn't break anybody, so it does not appear to increase the risk of egg allergy. So I think it's a great idea, though. I mean, I give them a lot of credit for going through this because it'd be nice maybe to look at an easier population with lower risk and see if we can, get some similar results, but apply it to a larger population. So I'm going to move on. My next study is about influenza vaccine, and I know Paul's got it.
How many of those were breastfed, Kevin? I'm sorry to interrupt. Where did they get the egg during the first 4 months?
So that's the question, and there was no discussion about breastfeeding as it pertained to egg exposure. But that's one of the questions: where these babies get exposed to egg early in life, to the skin? Is it breast milk? And I think it may be breast milk, maybe several exposures.
I missed something. Did they check IgE levels like at birth?
No.
I just wondered if you follow them prospectively, then you might have an idea.
I was impressed they could enroll kids at 4 months of age.
Yeah.
I'd be curious to know, though.
Yeah.
Impressive.
Kevin, what do you think is the incidence of anaphylaxis and eczema in the same kids? I mean, you're talking high risk for eczema, but do you also think they represent high risk of anaphylaxis too?
It would seem so, especially with a lot of sensitization and higher IgE levels.
Do you see that?
Oh, do I see it? No, not really.
I don't see many people with anaphylaxis and eczema. It's either one or the other, it seems like. So that's sort of an odd group to pick.
Yeah. I mean, eczema, high specific IgE, for sure, or even total IgE, but not anaphylaxis. They got really unlucky, I think, or something.
That's just a strange group to study.
Well, you look at, statistically, you look at the kids that fail, baked egg challenge. In other words, the same amount of egg protein. It makes you wonder, maybe they need to start with lower doses. Yeah, nobody knows, but like micrograms.
That's the whole problem with food allergies. Paul's question: why do some people get eczema? Why do some people get anaphylaxis? Why do some people have positive skin tests and they're clinically normal?
Yeah.
So just the markers we have of how much IgE you have and what your skin test is doesn't tell you what the clinical outcome is going to be.
Yeah, and you get an IgE of 20,000, and those kids almost never anaphylax. They just have terrible eczema, and it's just there's a skew in that population that's really odd.
Did the IgG4 correlate with any statistics about adverse reactions?
They didn't tie those two together, actually. They did link it to the IgE, so the IgE level and a ratio, but that wasn't actually drawn back to the anaphylaxis risk. I think they were caught off guard, actually, to have gotten the anaphylactic events, to be honest, and I would have too. I would have expected it. So looking at seasonal influenza vaccine and egg allergy, I always had to re-educate myself every year. So influenza is a horrible disease, hundreds of thousands of hospitalizations every year, tens of thousands of deaths. Recommended yearly vaccination if you're 6 months or older, particularly if you have health conditions. So kids who are 6 months to 6 months to 8 years get 2 doses the very first time they get the vaccine. Starting in 2010, the strains all include H1N1. And I guess the wrinkle for us is that these are grown in embryonated chicken eggs or hen's eggs. And so it's not really required that the manufacturers list the low albumin content, although in this table over here, you'll see that these different manufacturers have different levels, all 1 nanogram or less, nanogram per milliliter. Some are strikingly low, so this Fluzone, which is a high-dose vaccine, is 0.1 nanograms per milliliter. So again, most of these manufacturers come in at less than 1 nanogram in the trivalent influenza vaccine. The live attenuated vaccine has even less. It's also a smaller dose given in the nose. And based on research in the 1990s, it's probably unlikely to have some form of reaction to the vaccine with these low levels of egg protein.
Nanograms or micrograms? You have micrograms.
I'm sorry, micrograms. I misspoke. Micrograms. These are from the vaccine information statements from the different vaccines. So in the trivalent, the TIV, this is an inactivated vaccine. There may be thimerosal, and I always have to remind myself, because parents always ask, is it thimerosal or not? It's going to depend on the brand, more or less. With the live attenuated vaccine, there is no thimerosal, but I wasn't really aware of this in my mind, or I had forgotten that this is actually indicated for age 2 to 49, so if you're younger than that or older than that, it's no game. And also because it's primarily aimed at kids, there's a lot of data about no kids with immunocompromised states, kids who are on high-dose aspirin, so probably the Kawasaki population, I'm going to guess. And then this vaccine has not been studied with respect to egg vaccine, so all of our discussion about influenza vaccine and egg allergy does not apply to the intranasal vaccine. So looking at the 2011 parameter that came out in the annals, their general theme was that the risks of not vaccinating were worse than actually the vaccine itself. So go ahead and find a way to vaccinate kids who have egg allergy, or patients in general. If you have a known egg allergy, it's probably best to get evaluated an allergist before receiving the TIV, the flu shot, and especially if an anaphylactic event to egg. No longer recommended skin testing with the seasonal flu vaccine because it really wasn't a very reliable predictor of the reaction to the vaccine. Does anybody still do skin testing here? Good. You're not supposed to. And but yeah, we're going to get to that in a minute. So there may still be a role for other kinds of vaccine allergy, picking up some other factor that might cause a reaction to the patient. So 2 choices. You can either give give the 1 dose and wait 30 minutes and be done with it, of course in a safe environment, or you can do still the 2-step. So you get 10% of the vaccine, wait 30 minutes, give the remaining 90%, watch for final 30 minutes. I remember back in the H1N1 days, we were saving the same lot for kids who need that second dose, and actually if you can just keep the same brand, that's good enough. So there's not enough variability between lots apparently that it's worth keeping one particular lot for the next time. So in 2012, the update gave even more reassuring data that patients with egg allergy can probably receive the trivalent vaccine. At this point, there's greater than 600 patients who've had the vaccine and have been saved with no serious reactions. A small percentage, so up to maybe 5 or 6%, have had cutaneous, mostly hives, respiratory or GI symptoms from the vaccine, but again, no serious systemic reactions. There are a lot of studies that didn't actually specify who was egg allergic and who wasn't, but if you're able to fold in those data, it's a very, very reassuring picture overall. So in the 2012 update, still no skin testing recommended for the vaccine for giving it, and patients with minor reactions can get their vaccine at the PCV office, or if it's a severe reaction, probably at an allergist's office. I'm going to skip the slide again. So the study I looked at was also in the In Practice journal from this past month, and knowing there's recent changes and updates in the vaccine parameters, how are we doing as allergists in complying with the guideline? So this was a questionnaire that came out from the QuadEI through email, and they had around 4,000 sent out and 895 replied. Anybody reply? Oh, right. Another person. Okay, we got a few people. Good. So 1% of allergists still thought that the flu vaccine should be contraindicated for patients who have egg allergy. So don't do it. 3.8% don't give the vaccine to any egg allergic patients in their practice. They just don't want to do it at all. 17% give flu vaccine to patients with only mild egg allergy, so they actually will stratify it that way. 13% perform skin testing still, or some form of graded challenge. And then 49% felt that it can be given safely in a PCP setting. it's a bit of a loaded question because, again, we know that if it's a minor reaction, probably okay, but if it's a major reaction, it's not a good idea. So they were comparing basically before and after the guidelines were published. In general, there was less intradermal testing. So now there was only 31% of allergists doing it, whereas before we were doing it 65% of the time. There's a lot less of the multi-step desensitizations. I remember doing those. Kids are not fans of those. 35% now versus 65% before. We're observing patients longer now, or doing it more often, and 79% versus only 21% before. So that's great to see. And then there are some who are still giving, I'm sorry, if you do a skin test, will you actually give the trivalent vaccine in spite of that? And most people will still. All these factors here are significant statistically. There was no reduction in the frequency of prick testing. So again, the top line was intradermal testing. That was different. So we are doing less intradermal testing, but still about the same amount of prick testing. The ones who are allergists who are not doing prick testing, that was interesting. So they had read the guidelines, they were in academic practices, and they had fewer years in practice. Those are the common bonds. These things kind of come out here on these tables. In table 1 here, just the demographics that are available for the respondents, the population. So a lot of them were in academics, had been active in oral challenges, 94%, so very busy, taking some risk, I think, in their clinics. Had attended the meeting in the past 3 years. What else? Well, that was kind of interesting to see. So here's some data looking at whether skin testing was necessary. Most allergists disagree, which is probably the right answer, I think. Some would say only if the egg allergy was severe with a skin test. Looking at the albumin content, some allergists felt that it was actually important to know that it was a low content versus high content. But I think the reality is that it's pretty low in general, and it's probably not going to be a factor. And this is kind of interesting. This group over here, or I'm sorry, the next slide. Over here, I advise my patients to receive TIV at their PCP office. How do I make this work? Where's Drew? There it is. Okay. So I advise my patients to receive TIV at their PCP office. And 17% of allergists said, you know what? Any patient can do that. It's a little cavalier, I thought, but I keep wondering if the future will look back and say, did anybody really react to this, or was it really a real threat? How many people agree with the statement that for any child or any patient with egg allergy can receive it at PCP? Would you be comfortable saying that?
I think PCPs are not going to be comfortable giving the shot to some people, so it's not a starter.
Yeah, exactly.
So anyway, I thought it was interesting, and we're all doing flu vaccinations, I'm sure. And I'm just sort of curious, has anybody had a reaction in their offices to patients who have egg allergy? I just never heard of it.
Right. I mean, from the Hopkins study, there's also no data to support the graded challenge. I mean, the outcomes were the same.
Yeah. The two-step or the...
The two-step.
Yeah.
In other words, both skin testing and that really made no difference. If somebody was going to have some reaction, they'd have it after the whole thing anyway.
And before this came out, I didn't even like doing the graded multi-step. Just like torture to the children.
Yeah. Well, there used to be like a 5-step, 0.05, 0.0.
Those data are probably better than the real world, because obviously academicians and more recently trained people would probably answer the survey. So don't really know what the general allergist is doing, who decided they don't know enough about this question to take the survey.
That's true.
My partner looked into this a couple years ago, and the companies say that their vaccines have less than a microgram, and actually most of them had less than a picogram. So this was like 10 to the minus 12 grams in most of the standard vaccines. And I think as their production gets cleaner and cleaner, I think this is hopefully going to die in the future, because there's just not enough egg in there to do anything.
Well, I didn't put the data up there, but the H1N1 vaccine when it came out, that 2009, it had so little egg in it. It was remarkably low. And you can tell that they really scrubbed up the process and cleaned out most of that albumin.
Now we supposedly have Flutrol with no egg, but we can't get any of it. So it doesn't matter if it's available, because you can't get any of this stuff to vaccinate everybody. Has anybody been able to get Flutrol at all? Does anybody care?
I'll just mention that the Red Book, the most recent Red Book, says that we don't need to test or do anything. Just give the
influenza vaccine.
I think that's where everything's headed, but we still get all the primary care docs, if there's any history of egg at all, they still come to us. And then the parents, you just ask them, do you want us to do it in 2 steps or 1? And most of them don't care. So
I get MMR questions from time to time. That was in the 90s.
Yeah, I think it might die among allergists. I'd be very surprised.
All right, you're up.
Oh, that's mine. That looked familiar. I was going to say.
Very suspicious.
Are they 2 separate files? Oh, were they in the same file?
I don't know. I was looking at on a file, so what file they're in is a whole different issue. That should be it.
Did another.
Yeah.
Yes, wrestle.
So along the same note with Kevin, I was interested in this because I thought this question had died a long time ago. But then I had 2 patients on the same day come in and say they didn't want to get their child vaccinated because of Guilan Barre. And I thought, was that even happened since the 70s? And it was sort of interesting reviewing this literature because it's ancient literature. But those of you who are alive in 1976 when we had the swine flu outbreak, there was a 7-fold increase in post-vaccine surveillance of Guillain Barre syndrome, and they sort of pictured that as an epidemic, and they had 45 million people they vaccinated within a few months during this swine flu epidemic. So the question of influenza vaccination, I thought, was sort of moot, but it's not. So if you look at influenza, there's thousands of deaths per year worldwide, a lot of hospitalizations, a lot of morbidity. And since we had this H1N1 and now the H5N1 bird flu, I mean, there's no way to treat that successfully, so vaccination is really the only option that we have. So if you look at the flu vaccines per se, they all have adverse events. So that's why the VAERS system was set up. That was why the VAERS system was set up by the pharmacology or the FDA and the pharmaceutical companies to deal with the adverse reactions. Because no matter what you do, there are some adverse reactions. But this H1N1 virus was the one that was supposedly going to kill us all from 1976 because it matched the swine flu, and then our last H1N1, it was the same thing. And neither epidemic ever evolved into anything, but the panic factor was actually quite high. So they gave, again, 45 million people within an 11-week period got vaccinated, and in the post-vaccine surveillance, they figured that 7 times more people who got the vaccine got Guillain Barre than people who did not have the vaccine. And it typically came within 6 weeks after vaccination. So there have been 10 studies that have looked at this data since 1977, and looking back epidemiologically, trying to figure out what's going on, but they still can't figure out what caused this association. And then it's sort of like Dr. Wakefield and his autism literature. This is just a sort of a cult following that flu vaccine causes Guillain Barre syndrome. So if you don't want to get a vaccine, you can either pick Guillain Barre or autism as your reasons for signing out why your kid doesn't get a vaccine. And there's still literature support, although not very good. So these 10 studies all had some methodology issues, but they're all pretty well done studies. 4 suggested that there's a small increase in the risk of Guillain Barre following vaccine, with a risk of 1.45 to 2.35, or 0.06 to 0.16 per 100,000 vaccinations. And they all show a risk that's a full order of magnitude less than the studies from 1976 swine flu vaccine. There's a couple of studies that actually suggest there's an increased risk of Guillain Barre following natural influenza. So like everything else, now we're talking about bee stings and egg and food allergies. This is right in there with the clean data that we have to look at, which just separates out very quickly. So the increased risk of Guillain Barre following a natural infection was higher than the risk following the vaccinations themselves. So this literature, if you really try and clean it up and you say, what's the natural risk of Guillain Barre versus what's the risk with vaccination, there's a higher chance you'll get Guillain Barre if you get the flu than there is if you get the vaccine to the flu. So that sort of changed the tenor of this discussion considerably, because now nobody thought anybody got Guillain Barre from the infection. They thought it was only from the vaccine. But if you look at the statistics, actually more people get Guillain Barre from influenza than they do from an influenza vaccination. So if you thought you didn't understand it before, now you can not understand it even more. So in light of this data, you can't make any conclusions because the data is too conflicting. So they did a study in Sense to look at it one more way, and that's the people who had a history of Guillain Barre after a flu vaccine. In England, they found 927 patients with a history of Guillain Barre, and 300 of them got subsequent flu vaccinations after that. And they reported that 11 of the 311 reported a recurrence of symptoms, but it wasn't full Guillain Barre, it was just weakness and fatigue after the vaccination. And of the 29 who got Guillain Barre with the vaccine 6 weeks prior, 2 of them had recurrence of their symptoms of Guillain Barre, again, at a much lower level. And then a second study of 100 people, 107 with Guillain Barre, they didn't notice any recurrences at all in that group. So again, there's 2 conflicting studies. So none of the studies on the relative risk of flu and the subsequent development of Guillain Barre allow a risk assessment due to the small size. And when Kevin flashed up his thing about vaccinations, if you have a history of Guillain Barre with your vaccine, it's only a precaution. It's not an absolute contraindication in the FDA guidelines. They don't even say you shouldn't have a flu vaccine now. They just said it's maybe a concern. Talk to your doctor if you had a history of Guillain Barre before. And I thought that was a fairly ironclad rule. You had Guillain Barre, you never got another vaccine. But that's not even in the language that the FDA puts on the vaccines now. It's just a relative contraindication to do that. So from reading this article, I can make absolutely no conclusions. And if your patients ask you, is there any risk? I think the best thing to say now is your risk of Guillain Barre is the same if you get the flu versus if you get the vaccine. But your chances of dying from the flu are much higher than if you get dying from the vaccine. So that's the only way you can really make any conclusions stick at all. And I just thought the fact that even with a history of Guillain Barre, the FDA says it's just a relative contraindication. They must be pretty comfortable with this data, that it doesn't show anything. But I had 2 people in the same day tell me they didn't want flu shots because of Guillain Barre, and I thought this argument was dead. So obviously it's not quite dead because 2 people asked me.
Is it even accepted that the 1 year of swine flu vaccine, that there was something different about that vaccine and the incidence of Guillain Barre was higher that one time? Or is that not true?
They've done 10 studies trying to look at the epidemiology, and they can't prove that there was really even a difference then. But, that H1N1 strain was the same as the one we just had, basically. And this last strain, we didn't seem to get much in the way of side effects from it. But no, they couldn't go back and prove anything with that vaccine at all. Although my sister and brother both got sick when they had that vaccine, and I was in grad school and I couldn't afford it, so I didn't get it. And they both got really sick. They didn't get Guillain Barre, and I'm like, oh, I'm glad I didn't get that. So now how do I get to the next one? Matt, are you our computer wizard today?
PowerPoint.
Yeah. That's a quick. So this is a blood test for nickel allergy, and I just was interested because this comes up now. I don't know how many of you are getting patients.
Paper or the PowerPoint?
I don't have a PowerPoint with this one, but Matt clicked it up anyway. So I don't know how many of you are seeing patients sent in by their orthopods that are going to have a joint replaced that now have nickel dermatitis or contact dermatitis from their jewelry. And this is becoming in our group maybe 3 or 4 a week now. It's got to be quite large between the 4 of us. And as all of us know, nickel's the worst offender, and it'd be really nice to have a blood test so you didn't have to do a test, bring them back again. So it sort of turns a 3-visit into a 1-visit. And the folks,
the other metals, though, that'll be the question, because you do all metals, not just nickel.
well, the only test that looking like the government is going to certify is this nickel. The other ones they're not going to look at.
So you're still kind of stuck, and they come in, you're still going to do the other metals.
Pretty much. Well, when we patch test them to all 12 metals. And we get it from the company in Canada because it's really easy to get their stuff. Allergies from Alberta. But, nickel's still the big issue. And I'm just frustrated because more people respond to the tape on the patches than they do to the substance. So we have built-in controls all the time because they react to adhesive. But anyway, this is a test that's actually getting pretty close to the government certifying. And they did 8 concentrations of nickel. And you're not going to be able to see this because I didn't make a PowerPoint. I just sent this through because I didn't know what was going to get to you on my PowerPoints. And they figured out that 50 micrograms per milliliter it's just standard lymphocyte stimulation assay, basically, is what they're doing. And if you use 50 micrograms per mil of nickel sulfate, it actually killed all the lymphocytes. So that was not a good test. So they settled on 0.1, 1, and 10 micrograms per mil. And they did what's called a positive stimulation index. And when you stimulate non-sensitized people, and this is the first study where they compared patch testing to this blood test. All the other ones, they just do the blood test and try and tell you that it's statistically significant. But if you use those three concentrations and you have a stimulation index of greater than 10, then basically it correlates with patch testing about 98%, which is pretty nice. So it finally looks like we may have a test for nickel that you could just do a blood test and send it off. Hasn't been approved yet, but the folks at Colorado are sending it to the FDA now to be looked at and see if they can use it as a diagnostic test. So in the near future, we might not have to patch test for nickel.
What's the usefulness of either of those tests in predicting whether people are going to respond to implanted metal
Well,
to show they have sensitized lymphocytes?
see, you're looking for an epidemiologic study, which would be really nice to have, but I don't think there is that now. It's mostly anecdotal stuff, and this one orthopedics group in Colorado said that 10% of their failures of prosthesis now are metal rejection, and that's at least the reason they're given for
That's what
having.
they believe.
Correct.
There's really no evidence.
When they take it out, they see stuff and they do biopsies of the bone, and it looks like there's infiltration of lymphocytes. But their suspicion is that they can't find a mechanical failure, they can't find a glue bond junction failure, and then these things are still all nasty when they take them out, and they're saying it shows that they're allergic. But I don't know, 10% seems like an awful high number for prosthetic failure in general, so they may be not such a great orthopedic surgeon in the first place. But being a biomedical engineer going into medicine, the interesting next step for this is they're trying to coat the prosthetics with bioglass, which would be really cool if they could figure out it's a hydroxyapatite preparation they can actually put on the metal. So they got rid of the glue. The first thing is they make fenestrated stems now so they don't have to hammer them into the bone and glue them in. Because the previous failure, you have three interfaces. You have the bone glue, glue metal, and then metal hip. So you have three places to fail, and it was almost always the glue metal interface. So they got rid of polymethyl methacrylate by making a fenestrated stem, and then the bone grows through the stem, so there's no need to anchor anything. But now they're still seeing this rejection issue, and 10%, I think, again, like Gary said, is quite high. But the orthopedics guys are getting pretty nervous about this now. So they'll usually say on the request, allergy testing for metal, patient has trouble with earrings. And that's usually the request that we get. And then the patch testing is, we had Hackett talk to us a few years ago about patch testing, and the derm folks say it's 85 to 90% accurate, but I struggle with that number. I'm not sure. I would say 85 to 90%, but then hopefully all our people are negative.
We have Mike Martin coming later in the year, the guy in the dental school here who's the local expert on this, so he'll update it from his perspective.
Yeah, a couple of years ago, that was a good talk. He gave a metal testing.
I always wonder about the metallurgy. I mean, are they keeping nickel out of these alloys, or is it sneaking in some way? Because if it did, that would be a problem. But
we don't have anybody here from Zimmer, which would probably be the company you'd want to talk to. But now, if you look at the alloy slip that comes with the prosthetic, they'll give you a percent. They won't give you a concentration anymore. They'll say this contains less than a certain percent of these different compounds. And obviously nickel's the one they want to get rid of the most. But it's now showing up as gold allergies, becoming more popular than it used to be. The cobalt's still there, nickel. So you never know what you're going to find. But it would just be nice if we could put an interface barrier there so that we didn't have to deal with metal into the bone. So that was one I was just going to put if none of my PowerPoints came through, but that one actually came through. So now how did you get to the other one?
I think you can just go forward. You have more.
I just have one more that I thought would give any. Yeah, this is the same thing. Just compares it to the blood test. So, let's see. That's Kevin's. Those two.
I don't think it came through.
Yeah. Darn. I had one more. Well, my last one was sort of because of industry. I don't have, but obviously my computer skills came through ahead of my actual, hopefully, medical skills. But there's a new FDA designation of a way to approve drugs, and it basically is a way to fast-track drugs that are in clinical studies. Oh yeah, I have that.
We have a printed copy. And
Yeah, I thought that one for sure came through. So
The first one on
it's called Breakthrough Therapy, and actually it was on the email that got sent to me, so I don't know where it went. Anyway,
at the end of one of these.
it could be. See here. Oh, there it is. Anyway, it's a new designation for drug development called breakthrough therapy at the FDA, and this has to be a drug with life-threatening illnesses and no adequate treatment available. And then the drug is looked at initially after clinical studies instead of waiting till they go through the whole series. The average time it takes from a drug to go from the first clinical study to patient use is about 7 years. So they have a couple pathways to get things through faster, but unfortunately those pathways haven't been very successful so far. So they made this breakthrough therapy pathway. And this is one that Congress passed in 2012. It's called the FDA, FDA-Asia is what it actually comes down to. It's the FDA safety and it's an FDA safety issue, and it was passed into law in 2012. And what it did is it enabled the drug companies to come up with this breakthrough therapy designation. And what that does is allows the drug company to have an alternative pathway to bring it to market. And it gives patients access to these drugs pretty much right away after the phase 1 clinical studies are done. And they don't have to wait till it's in phase 3 or compassionate release. So the reason for this is that the people at the FDA are saying we now no longer have these drugs that are just, we don't know how they work and we don't know what they do. The real reason for this is because of now the molecularly targeted therapies, where they'll come in with a single step in the treatment and they know exactly what the drug does and how it does it. So they're trying to say that if we know that enough about the drug up front, is there a reason that we have to go through these long studies to just gather data like they have in the past, where they had drugs that they didn't understand the mechanism of action, they didn't understand how to dose it. So now the drug companies have sort of made this different because they bring a drug to the FDA and said, this is what it does. It blocks this step in this replication cycle. And then what they'll do at this point is they go through the first clinical studies, and if the drug looks really favorable after the first clinical study, then the company can petition to put it into this breakthrough therapy category. And what that does is it basically gets the FDA to align with the drug company instead of the adversarial relationship they've had in the last few decades. And the FDA has them do specific studies to allow rapid approval of the drug in the market. And it has to be human studies. In the past, these other processes, they would accept animal data, and the FDA says, no, we're not going to do that. We have to have human data before we'll even consider this. And in Section 902 of this FDA Safety and Innovation Act, there's 2 criteria that you have to get this drug through. The first is it can only be used to treat a life-threatening disease or condition. So it can't be another treatment for acne or another treatment for, group A beta strep. It has to be a specific indication. And the second is you have to have clinical data that says the drug is more significant than anything else. And you can use that as an endpoint in the study, just that it's better. And you don't have to go through the next 5 or 6 years worth of trials. So what the FDA does then is they sit down with the manufacturer and they say, this is the data that we want. And they tell them what study to do. They help them organize the study to get the data the fastest way. And then they'll work with them all the way through. If there's any study hangups or study problems, it becomes sounds like a very symbiotic relationship between the FDA and the manufacturers now. Instead of what it used to be, it was a very antagonistic one. And if they didn't like you, they could hold your drug up for years just because they didn't like you. And it sounds like they've come to grips with the fact that now the drugs are coming to the FDA for approval, and all the bugs have sort of been worked out now, except for the clinical data in humans, and they're trying to push that through so that the patients can decide on informed therapy. And they want to stress that this isn't a drug approval; it's actually just a process that lets the drug go through the FDA faster. So they accept the fact that not all these products are going to end up being breakthrough therapies, because when they get into the larger human studies, they'll find that they don't work or there's unexpected consequences. But that's why they want you to realize it's not a drug approval. but they're going to allow publication of these lists of drugs. And the list of drugs, they've had 80 requests for breakthrough designation in the last year and a half, and 26 have been granted. So this is a really new fast-track approach for the drug companies to get the drugs through the FDA, but at the same time, what it also does is it gives the patients access to the drugs as soon as the first round of clinical data is available. So instead of trying to recruit people in one research site around multiple research sites around the country, they're letting this be a more centralized approach to getting the data quickly. And then the old way was called the fast track, and that was done in 1997. And basically what it did is it shortened the process by letting the companies give the FDA parts of their research data along the way instead of waiting till the study was complete and then have the FDA look at it. And that basically shortened the FDA approval process by 4 months. So really wasn't a fast track. And then the other one.
Those 26, that means 26 drugs have come to market, or they gave 26 applications the right to go through this process?
Yes, 26 applications. They tried to get 80 requests through for breakthrough designation. Because, again, what that does is it aids the company in getting patients because it allows them to recruit for the studies. And the FDA lets them recruit as a study drug. It's not just compassionate release you have to hear about and ask them yourself. And they only granted 26 in the last year and a half.
Nothing's come to market through this process.
Correct. And it's not a drug, it's not a drug approval. It's actually just a way for the patients to get access.
Are these brand new drugs or drugs that may have been used somewhere else in the world?
I think about 90% of them are brand new drugs, but there's a couple where they look at optical isomers of previous drugs, or they look at combinations of 2 previous therapies. But it was, after working trying to set up a research center in our clinic and working with the drug approval process, it's a pretty onerous task. And, a lot of the drugs now are coming with so much extra data that the FDA sort of said, listen, we really don't need these just general studies for safety and efficacy. We really need to look at the initial data, select out a population of people where we get the answer the fastest, and then bring it to market as quickly as possible. And it just seems like the pendulum has swung back the other way. Instead of we're going to protect everybody like thalidomide, now it's like, you know what, we're going to see if this drug works and we're going to help the FDA. Is going to help you get it to market if it's a good drug. And since we have so many people back there sponsoring our breakfasts and, telling us about these drugs, it's just nice to know that the FDA has changed their approach to this, and they're actually going to try and help them instead of hinder the process. And I just thought that was pretty cool.
Did they say how they define if a disease has a mortality, dangerous, life-threatening disease?
Dangerous and life-threatening are not really the criteria. It's just, it has to be something that there's no good treatment for. So whether it's, how you define the disease, it's more important that there's no effective treatment for it. And they won't let, in these studies that are done, they don't make them do placebo trials now as part of this new approach. You don't have to do a double-blind placebo study for one of these big new drug names because they know that they're going to put the patients at risk, the people who have these bad diseases. They don't think it's ethical to put them through a placebo-controlled challenge. So it sort of has changed the way they do the research now, because if you have some disease with a 98% mortality rate and there's no drug available, you put them on placebo and they're going to die. So it's sort of, I think they're just looking at the whole approval process.
Is there anything in our field that's going through this process?
Yeah, if this thing had made it through, there's all kinds of stuff. There's the 23 that got the new designation were printed on the PowerPoint that didn't come to you. But most of them are neurologic drugs and infectious disease drugs. There's not a lot there's 2 for asthma that I know about in that group. Out of the 20, there's only 2 that I saw were something that looked like we're going to use it. There's a lot of chemotherapy stuff and a lot of there's some cardiac stuff and chemotherapy drugs, but we're not going to see most of them. Rheumatologic drugs. It's sort of where the research is now and all those biomolecules that they're using now. But it was a neat thing to see that they're trying to streamline the drug approval process, and I fought with them for so long, so many times and lost. It was just nice to see. So,
all right, that's a lot of material for 1 hour. Next week, we have a guest speaker, Dave Skoner from Pittsburgh, who's expert in steroids and growth in kids. So we visit that issue
David's in there. Anyway, her IgG total has not gone up this side. But
Yeah, that's interesting. Oh, okay. So you go in and do it that way.
because she's, and then she's got I don't think she can make it tomorrow. She's patient.
You're guinea pig.
How quick can they get him started?
Well, that's the thing. They were apparently a series of court of law. But a jury of our peers has not. They're not our real peers, they're kind of our peers.
Even though they're not there.
Are you going to celebrate Hanukkah or Thanksgiving? No. Why is Hanukkah so early this year?
Anyway, so she thought she had to leave it.
Oh. But this is really early though, right? Usually it's after the 1st of December, isn't it?
You did that whole thing on the egg and the vaccine. I thought that they just come out at this point and said, you know, anybody can make a vaccine. The red ones, no, and that's what that's you don't have to worry about it.
But she's a nice.
There were some chairs.
She's one of these kids that doesn't look all that.
That's what I've heard too.
So that's what I've been telling
So
everybody.
maybe not equal, that's why I said too long, you can send them away.
No, you're
He said, you know, sometimes they look good.
I was out of work for a while.
Dr. Wright, I didn't want to interrupt it. Well, I the computer. It did, and I think what happened was that there were actually presentations, so
Yeah, 4 of them were mine, and then there was like 2 of Lynn's and 2 of Kevin's, so this picked up
so that's what the thing was, is that we just didn't drag them all over.
Oh, no, that's okay.
The 2 of the, how many did you
I sent
have?
4 in.
Drew those
Actually,
Yeah.
I was hoping
So-
to get 2 to you, and I got all 4.
Yeah, exactly.
She came over, set up, and went back
Yeah, there were
home.
probably like
Oh,
12
God.
presentations that both of us were trying to decide which ones to drag over, so we just drug the PDF on. It was,
not a big deal.
I'm sorry, I hope
No, no,
that
no.
didn't
We're just getting old school.
impede your
No.
creative process there.
My creative process was desperation yesterday. Can you quick send this? Oh, sure. So I would give it to somebody, and my nurse would go, oh, it's done. It's like, okay, cool. And I come back, what about these 2? Oh, I forgot those 2.
Wait, your nurses know how to use the internet?
Oh, gosh.
My nurses know how to steal things out of literature and paste them into an email.
Wow. Ours are really good at Facebook.
Yeah.
It's exceedingly good at Facebook.
Our clinic shut down all Facebook access. You can't get on that site at all. They found that during the day, 30% of our CPU time was used on Facebook.
Oh my gosh.
So the administration says, okay, that's not going to happen anymore. So you can't get on YouTube, you can't get on Facebook. Only certain people can get on Google. So I mean, all the social media is just trash. So I thought that was pretty funny.
That's tragic.
Tragic.
What kind of horrible employers are you?
Can't get on Facebook.
Are these your gloves down here? Are they yours? Or are these somebody else's? Are these your gloves, Mary?
No, they're not. I was kind of eyeing them though, thinking, wow, those look pretty nice.
Okay, so he wasn't like fumbling, stumbling, so I was like, okay, I'll just like keep quiet.
Oh, well,
Yeah.
they enrolled me.
Yeah.
Just
take it. Well, I was just gonna just take it back to the allergy lab.
You know, they didn't really comment on the legality of the thing. What they're really trying to do is now that patients know all this stuff online, what's happening is they're trying to get into these studies really quick, and they don't want to go to Washington, D.C. to get in a study. They want to be able to get in a study locally. So now the drug companies are, they're providing the drug as long as they fit the protocol. What's really changed is there's no phase 3 approval. You don't have to do the 2,000 patient protections against standard therapy, because most of these drugs only get approved for this category if there is no decent therapy available. So you can't give them a placebo trial if they have a fatal illness. And say, well, we're going to kill 20% of you just to make it a scientific study.
So like in our field, would like say maybe like a Sunrise fall into this, or I think what else? Like a replacement product would be easy to.
Yeah, like icatibant would be one that they would fit into this because the standard therapy doesn't work. So can we just use this instead?
Let's say an anti-L7. That would be. I'm very nervous giving it to somebody not knowing.
Well, but if you look at the studies that these guys had to go through with Xolair to get through the FDA, they could have shortened that by probably 8 years by doing
You're
this.
talking CLL stuff?
Yeah.
With all the safety data out there and everything else, yeah.
So, but they're saying, they're not going to look at any animal data, or it's got to be human data, but they're going to look at it after phase 1. And if the phase 1 data says, you know what, it works and it really looks good, they're going to blow them into this pathway.
So it's easier enrollment, it sounds like, more than anything.
Yeah, and instead of using a compassionate release for 10 years, if you know about the drug at all, they're going to open it up and say, this is the study that we need.